Hi, my name is Billy Rao, and I'm a bioinformatics applications scientist at PacBio. Today I'm going to share with you a bioinformatics workflow for comprehensive detection and prioritization of variants with PacBio HiFi reads. Over the past few years, we've demonstrated that HiFi reads are a powerful tool for discovering genetic variation. The read length allows them to be mapped to more parts of the genome than short reads, and the read quality allows for the accurate detection of single nucleotide variants, small insertions and deletions, and structural variants. The next analysis goal was to develop tools using HiFi reads to identify variants of interest to rare disease researchers. To address this, we collaborated with researchers at Children's Mercy Kansas City and Radboud University Medical Center to sequence and analyze more than 100 samples, representing 80 singletons and 7 trios that were negative after short read whole genome sequencing. For this collaboration, we wanted an analysis workflow that accepted HiFi reads as inputs and produced annotated list of rare variants as outputs. The general design has two main branches. On the left side, we align HiFi reads to the reference genome GRCH38, and we use deep variant, PBSV, and Tandem genotypes to call variants. We use tools like BCF tools, SleeveR, and SVPack to annotate and filter these and generate a list of candidate variants. On the right-hand side, we create a Denovo assembly of HiFi reads using HiFi ASM, which we can then align to the reference for variant detection and inspection of complex rearrangements. Before variant calling, we generate some descriptive statistics and perform quality control at the run level. First, we calculate some statistics about each run, like the yield, read length distribution, and read quality distribution. In this example, we were creating libraries with an expected read length of approximately 15 KB, and we wanted a median read quality above Q30, or 99.9% accurate. Next, we calculate some quality control metrics to guard against sample swaps. On the left, we look at ratios of coverage depth for autosomes and sex chromosomes to infer sex and compare this to the reported value. For example, in males, the X to 2 ratio is usually around 0.5, and the X to Y ratio is usually around 1. So they tend to cluster over here in this group on this graph. For females, the X to 2 ratio is roughly 1, and the X to Y ratio is very high since they typically don't have any alignments to the Y chromosome. So they tend to cluster over here. Because each sample was sequenced on multiple SMART cells, we developed a tool to confirm that 2 SMART cells were loaded with the same sample. On the right here, we use a k-mer-based approach to check for sample consistency between SMART cells. We count k-mers and compare a subset of these between SMART cells. 2 SMART cells from the same sample should have more shared k-mers than private k-mers. Every point on this plot is a comparison between 2 SMART cells in a large data set. Comparisons between 2 SMART cells from the same sample have very few private k-mers, but comparisons between 2 SMART cells from different samples have a higher percent of private unshared k-mers. Once we're confident with the quality, we move on to small variant calling. HiFi reads are aligned to the reference with PBMM2, our wrapper for the Minimap2 algorithm. Small variants are called using deep variant, producing a VCF and GVCF for each sample. For cohorts, we can joint call from the deep variant of GVCFs using GLnexus. Over a cohort of 80 samples, on the right here, deep variant calls a median of over 4 million single nucleotide variants and over 900,000 indels per sample. And we know that these small variant calls are highly accurate. When we compare both short read WGS and HiFi WGS single nucleotide variant calls against Infinium screening microarray results for the same samples, we see that HiFi WGS has higher sensitivity and specificity than short read WGS, here in the yellow, for most samples. Heterozygous small variants can be phased by WhatsApp using the long-range information from the HiFi reads in a process called readback phasing, allowing us to stitch together long haplotype blocks. This information can be added back to the alignments as well, allowing the user to sort or group alignments by haplotype visualization. In the example on the right, we're looking at a region of chromosome 22 that carries CYP2D6, along with its two highly similar pseudogenes, CYP2D7 and CYP2D8P. Using HiFi reads, deep variant and WhatsApp generate haplotype blocks that completely phase this 51 kb region. But these phase blocks can be much larger than a few genes. The CYP2D6, 7, and 8 region from the previous slide is bounded here by the red box, but this phase block, is denoted here in this blue line at the bottom, actually covers this region stretching for nearly half a megabase, allowing the user to assign variants to haplotypes even when separated by hundreds of kilobases. So you can phase variants over multiple genes. Over a set of 80 samples, the mean WhatsApp phase block size was 205 kilobases, and the phase in G50 was 389 kilobases. Once small variants are called and phased, we provide tools to annotate, filter, and prioritize variants of interest. First, we add the population frequency from 71,000 short read sequencing samples in the Nomad database and 40 HiFi sequencing samples released by the Human Pangenome Reference Consortium. Then, if multiple samples are available for a cohort, we check whether the variants fit within the expected mode of inheritance, and we annotate consequence using BCF tools. So we annotate whether the variants interrupt an exon, interrupt start codons, their frame shifts, etc. And we identify potential compound heterozygous pairs using SLEVAR. And then finally, we generate a list of candidate variants with a score for each gene based on how well the gene matches the known phenotypes. Annotation and filtering greatly reduce the number of candidate variants. In the chart on the right, for a representative sample with approximately 4.6 million single nucleotide variants and 1.2 million indels, we can see that filtering based on frequency in either Nomad or HPRC reduced the number of candidate variants by roughly a factor of 10. Filtering based on whether a variant impacts a coding region will reduce the number of candidate variants by roughly a factor of 100. And filtering based on both of these together will reduce the number of candidate variants by a factor of approximately 10,000, resulting in hundreds of S and Vs and tens of short indels. For structural variants, we have a separate analysis workflow. We can use the same PBMM2 alignment generated earlier for small variant calling, and we call structural variants using our internally developed tool, PBSV, to generate VCFs. For multi-sample cohorts, PBSV can perform joint calling as well. HiFi WGS with PBSV detects approximately twice as many structural variants as short-read WGS approaches and can detect copy number neutral variants like inversions that are difficult to detect with short reads. In the example on the right here, PBSV has detected a 407 base pair heterozygous copy number neutral inversion containing an exon. The exon denoted here by the small blue bar. A color change on a row indicates a change in alignment orientation that you would expect to see for an inversion. So in the example here, we have an alignment to the top strand, which flips to an alignment on the bottom strand, showing the inversion, and then flips back to an alignment on the top strand. Similar to our approach for small variants, we annotate and filter the structural variants to generate a list of candidates. First, we annotate and filter based on population frequency from 3 sources: 14,000 short read sequencing samples from Nomad SV, 40 HiFi sequencing samples from HPRC, and 15 PacBio CLR samples published in Denovo 2019. If multiple samples are available for a cohort, we can check whether the variants fit with expected modes of inheritance. Then we annotate with functional consequence using an internally developed tool, SVPack. And finally, we generate a list of candidate variants with a score for each gene based on how well the gene matches the known phenotype. Again, annotation and filtering greatly reduce the number of candidate variants. In the chart on the right, we can see that for a representative sample with around 24,000 structural variants, filtering based on the frequency in HPRC dataset reduces the number of variants by a factor of 10. Filtering based on presence in Nomad SV or Denovo et al. 2019 also has a small reduction. In combination, we're still seeing a reduction in the number of candidate sites here. If we filter based on whether a variant impacts the coding region, we see a reduction of roughly a factor of 100, or well, actually, it's closer. It's more than a factor of 10. And then filtering on all of these together reduces the number of candidates by roughly a factor of 1,000, resulting in tens of structural variant candidates. In addition to small variants and structural variants, we've recently added support for detecting short tandem repeat expansions at known loci. We're able to add this using the Tandem Genotypes tool published by Mitsuhashi et al. In 2019. This starts with a targeted realignment of reads at a set of provided tandem repeat loci, followed by genotyping and visualization. On the top right, I show examples of the output of Tandem Genotypes tool for three loci in the HG002 sample. The tabular output has fields describing the position, the repeat unit, the locus, and repeat unit counts for forward and reverse strand alignments. These are counts per read. Negative counts indicate a loss of repeats relative to the reference, and positive counts indicate a repeat expansion relative to the reference. In the graphs in the bottom, the x-axes are the count of the repeat unit, and the y-axes are the number of reads supporting each repeat unit count. So, as an example here, at the RFC1 locus, we have a pentanucleotide repeat motif that's intronic, and we see two alleles supported by the data. One is a reference-like allele. I believe it has one repeat insertion, and the other one is an insertion of more than 100 copies of the pentanucleotide repeat. The data are generated for a curated set of 45 loci from Mitsuhashi et al. 2019, but additional loci can be added. As an orthogonal approach to alignment-based variant calling, the workflow supports Denovo assembly. An assembly is generated by HiFi ASM, which we can then align to the reference with Minimap2 and call variants against using HTSBox. Over 80 samples, HiFi ASM generated assemblies with approximately 3.07 gigabases in size, with 28.1 megabase primary contig in G50s. 94% of the genome is phased, with phase block N50s of approximately 429 kb. Using the alignment of the assembly to the reference, we can more easily understand complex rearrangements like this 9 megabase contig, shown here, that shows the complex translocation involving chromosome 9 here in brown. This portion aligns to chromosome 9 along with an inversion here, as well as multiple regions of chromosome 2, shown here in orange at the end of the read. In addition, by using the tool HTSBox, we can call variants from the alignment of the assembly to the reference. The size distribution of HTSBox small variants closely matches that of deep variant. This is a graph of small variants bin by size. And the size distribution of HTSBox structural variants closely matches that of PBSV. So this is a similar distribution for variants that are greater than or equal to 50 base pairs, bin by size. So that's the current state of the workflow, but we're refining and adding new features. The version I've described is implemented in the Snakemake workflow management system using Conda and Singularity to manage dependencies. And we've written it with HPC job scheduling in mind from the start. So we have examples of scheduling parameters for Slurm and SGE clusters available in the GitHub repository. The Snakemake implementation is broken into 3 steps. Process smart cells, which performs run level quality control and aligns data from individual smart cells to the reference. Process sample, which acts on multiple smart cells from the same sample and performs variant calling and Denovo assembly. And process cohort, which performs joint calling, filtering, and annotation on cohorts of 1 or more samples. The code is available on GitHub at the at the URL at the bottom of the slide here. And we've got a lot of people to thank. I'd like to acknowledge the work of many PacBio employees that has gone into creating and testing the workflow, as well as the contributions and feedback of our collaborators at Children's Mercy, Kansas City, and Radboud University Medical Center. I'd also like to thank the Human Pangenome Reference Consortium and the group behind Nomad for the population data sets. And I'd like to acknowledge and thank the open source bioinformatics community that has made work like this possible. We really wouldn't have been able to do that without the tools listed on this slide. Next, Roberto Lleras will tell us more about a WDL implementation of this workflow that he and others at Microsoft have been developing to allow users to analyze HiFi WGS data in the Azure cloud.
Thank you so much, Billy, for the warm introduction. It's great to be back, and I'm happy to be here representing Microsoft to present to you our work in collaboration with PacBio on enabling the whole genome analysis workflow on Azure. Particularly highlighting some of the work we did around containerization, optimizing this workflow for the cloud, and data management. So you might be wondering, who is Microsoft Biomedical Platforms? Well, this is a recent rebranding that we've undergone from the Microsoft Genomics group, really reflecting the broadening of our scope and mission, looking beyond and including not just genomic data, but multi-omic analyses and building a cloud-based platform for enabling research as well as data analysis. Ultimately, Microsoft strives to enable genomic workloads at all levels, starting at the research and discovery phase by building and supporting open source and community-based tooling that enables data science and genomic analysis at the R&D level on Azure, enabling users to then take those workloads and automate and scale those to production levels when they have a locked-in workflow through building out and supporting workflow management platforms on Azure. We've enabled a number of those, and we'll be utilizing one of those today as part of our collaboration. And lastly, building turnkey services for clinical genomics that are secure and compliant for being able to work with the most sensitive of healthcare data. Our open source solutions enable us to build a genomics workspace on Azure that contains the ability to process and scale workloads of all levels, integrate public and host public data sets, interact with your data at a very intimate level with notebooks and tools that are optimized and built specifically for genomics and data science, as well as enabling data analytics to integrate genomic data with other types of information to do machine learning and artificial intelligence processing of the integrated data sets. All of this built on top of a secure and compliant environment provided by Azure, leveraging the plethora of tools and services built on Azure. One such platform that we've leveraged that is as part of this collaboration is our Cromwell and Azure workflow execution management engine. The Cromwell, as you may be aware, is a workflow execution management engine built by the Broad. And what we've done is we've built a specific deployment and runtime environment that is a GA for GH compliant to allow for either storage-based activation of workflows and automation of processing workflows through Azure Blob Storage, as well as through REST APIs, and submitting those workflows for processing through Azure Batch to low priority nodes by default. And by doing so, we can minimize the cost of those workflows while enabling them to scale both horizontally and vertically as needed for the particular task at hand. And for this particular workflow, there was a number of challenges that we built and accounted for as we're building this workflow to scale. So the PacBio rare disease whole genome sequencing workflow that we have enabled in WDL is both fast, cost-effective, as well as scalable. We've made it highly customizable and adaptable to different use cases, where you can run either a single smart cell of data as you need it, an entire sample, or multiple samples of data, or even an entire cohort at once, with simply a single initiation of a workflow. The workflow is well documented, including example data that's provided via our Azure Open Datasets Genomics data lake. And you can simply modify that by forking the repo to your own GitHub account, set up Cromwell in Azure, which is a single download and single executable to deploy, one command to configure the entire environment. You can then upload the example trigger file that's provided in the repo account and start customizing the workflow to your specific needs as needed. To enable this to happen, we've done a few things. First and foremost, we spent a lot of time looking at the extremely diverse tool chain of open source tools that are utilized by the workflow. And what we've done is, instead of building Docker images for every specific individual tool in the chain, we've actually built a modular Conda Docker image where you can simply specify your tool as part of the specifications input for the workflow. And you can build the Conda environment for that particular tool and access it in real time at the task level for your individual run of that workflow. This enables you to quickly optimize, add new tools as they become available in Conda, and update versions and tooling with very minimal changes actually needed to the existing WDL architecture. Moreover, we've taken all of the Docker images that we have written and we've brought those into the Microsoft Container Registry. So you're no longer having to rely on those and being available in Docker Hub, but you can also pull those in directly from another container hosting website. Secondly, we've made some modifications into the how the workflow handles input and output files, given the nature of running workflows in the cloud, which by default doesn't have shared file systems. So with Cromwell in Azure, we currently utilize blob storage for the main working directory. And the reason why we do this is that it's extremely cost effective. But this means that we have to do a fairly significant amount of copying data as we move through our workflow. This also enables us to do and enables us to utilize shared access signatures to quickly add and remove locations of data. So you no longer have to copy your data to a particular storage location to be able to access it. With this workflow and enabled by Cromwell in Azure, simply bring the compute environment to your data and you're off and running, while maintaining the security and compliance that you need for your data. So here are some preliminary benchmarking and cost numbers. When we run this data on 30-fold coverage of whole genome sequencing HiFi data for HGO2 on our West US region, we see a cost of about 45 cents a smart cell to prepare a smart cell for running. So in a typical 3 smart cell sample use case, it's about $1.45 to $1.75 a sample to prepare your data for analysis. The total cost of actually generating your complete set of variant calls is approximately $20 to $30. And these samples can be run in parallel, one or many at a time. However, we would advise that you, and will provide guidance on increasing your quota to be able to scale horizontally for cohorts larger than a trio. As part of that cohort analysis, We're still doing some final benchmarking and specifications, but we seem to observe that for joint calling a cohort, it's an additional $2 to $5. To generate all this data, it takes about 90 minutes of total wall time to process a smart cell, and you can process many smart cells at once. And to process a entire sample, it takes about 1 to 1.5 days. However, you can run several samples at once as long as you have the provided quota needed to scale vertically as necessary for your cohort. Now, we think that there's multiple ways that we can quickly reduce both cost as well as the time to result. And these are two key features that we are actively working on implementing in the near future for Cromwell and Azure. First and foremost, as we are currently using blob storage, and blob storage enables us to be able to reduce our storage cost, but at the sacrifice of having to copy data. We are moving to utilizing our new NFS on blob functionality. This will enable us to still utilize the cost effectiveness of blob storage while being able to create a shared file system. So you no longer have to transfer and copy files when moving from one virtual machine to another. We think that this will improve runtime and overall cost significantly. Secondly, we are building out the capability within our Cromwell and Azure implementation to be able to specify task-specific SKUs. And particularly, we found this necessary for deep variant, where we need to be able to specify a very specific virtual machine to enable the most optimal performance of deep variant. So with that, I thank you very much for inviting me to present, and I'd like to hand the stage back to Billy. Thank you so much.
Thanks, Roberto. We're really excited here about enabling our customers to do HiFi WGS analysis in the Azure cloud. If you'd like to learn more about how HiFi reads can be applied in genetic disease research, check out Jenny's on-demand presentation. And for more information about the sample and library preparation workflows for HiFi WGS, you can hear more from Christine's on-demand presentation. And if you need more information or you have any additional questions, please visit the links on this slide. Thanks for your time.